So this is National 5 Biology, Unit 1.2. This is transport across cell membranes. I would say for this there's quite a few practicals within the slides. Some of them you could actually do at home, and I'll talk about that when we get there. But what I would do is I would maybe split this into two sessions. So maybe the first 4 lessons you could do in a 1 hour and then have a break and then come back another day and do the rest of it. Okay, but as we go through it, just pause it whenever you like. And yeah, this is Unit 1.2. So learning intentions, you'll know about the cell membrane and the fact that it controls what enters and leaves cells. But we're now going to look at the structure of it and some new words, a couple of new processes: passive transport and diffusion. So describe the processes of passive transport and diffusion was the second learning intention there. Now, a couple of films just to start us off. So if you go into Glow, log into Twig and have a look at that one, the cell membrane, and then we've got a wrap. Yes, so go into YouTube and Google that, cell membranes wrap. Have a look at them and then come back here. So this is our animal cell, and cell membrane is what surrounds the cell, and we know that we have them in fungi and plant cells as well. There it is. And this, if you actually could zoom in, is what it looks like. Okay, so it consists of two parts, and those are phospholipids. So you can see they're drawn as little circles. Okay, 2 rows of them, and then spanning the membrane, or sometimes not quite spanning it, you also have proteins. These proteins do various different jobs, might be receptors, for example, or they might be channels to let things go back and forward. But that is your two parts, phospholipids and proteins. Now, cell membranes are called selectively permeable. Permeable, whether something is able to go through it or not, and what they do is they will only allow certain substances through. And for example, if you've got, say, a channel here which is a certain diameter, a certain size, then it may. Just to show you some other little drawings you could have. So there's your phospholipids up here, 2 layers, and here is your membrane. Okay, and this one's got a channel. Again, just another sort of drawing. You can see the channel down here, and here's a kind of simpler one. Okay, there's a phospholipid bilayer, it's called. This one, we don't have any proteins in that particular section. Just to show you, this is what they look like. Now, in order for movement to occur across membranes, you need something called a concentration gradient. And a concentration gradient is simply the difference in concentration of a substance between two areas. So if you imagine in this diagram on the left, that is the cell membrane. On the left-hand side, you've got a high concentration of this red atom or substance or whatever it is, and on the right-hand side, you've got a lower concentration. So a concentration gradient exists here. And if that is depicted in a graph, you can see there's area 1. This is the increase in concentration of molecules. So you've got an increase in concentration of molecules. So in area 1, it's higher, and in area 2, the concentration is lower. So a gradient exists between these two areas. Okay, and another couple of videos for you to watch. We're going to look at, remember the second learning intention was diffusion and passive transport. So I want you to have a look at these, diffusion through Twig, and then YouTube. If you just search for cell membrane passive transport, this gives you a nice animation of what is actually going on. Now, diffusion, and we talked about what a concentration gradient is. Diffusion is when molecules move down a concentration gradient. and that means they are moving from a higher concentration to a lower concentration, and this process continues until they're evenly spread out. So that is diffusion. So if you look at these molecules here, they're all concentrated in the top left area within this like a tank of water or whatever it might be, and you can see they begin to spread out. So they're moving to an area where there's a lower concentration of these molecules, and this process continues until they're evenly spread out. So that's diffusion. And if we look at cells, we've got here's O2, so oxygen's an example of diffusion. Around this cell, you can imagine it's in your, for example, in your lungs, you've got a higher concentration of oxygen outside the cell. So generally, you can see there's some oxygen going the other way, but oxygen is moving down the concentration gradient from a higher to lower concentration into the cell. Now that second term that we mentioned was passive transport. Now diffusion is known as passive transport. The word passive you might be familiar with, so I suppose it's the kind of opposite of energetic. If you're passive, you don't have to, you're not exerting any energy, and that is why diffusion is known as passive transport, because it does not require energy. It just happens naturally. So if you've got a higher concentration of a substance in one area, then it will naturally move to an area of which there's a lower concentration. So another cellular example here, you've got a higher concentration of glucose outside the cell, so it's going to move and it's going to move into the cell where there's a lower concentration. And the purpose of it, well, it's to allow important substances, for example, oxygen or glucose, into cells, and also you will have waste products accumulating within cells, for example, carbon dioxide. So diffusion allows these waste products to get out of the cells, to be excreted, breathed out, whatever happens once they've left the cell in order to remove it from the body. So here's a little task for you to try. So you can, when we're in the classroom, we'd ask you to do this in the table, but what you would do is draw the images below, and you can do this in a bit of paper just now, and then show what would happen after an hour. So press pause, and then we'll have a look at what you should have. Right, so we've got glucose on the left-hand side, we've got our cell membrane, and actually quite a good idea here is to count the number of glucose molecules. We've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Okay, so after 1 hour, what you should have is basically 7 on the left-hand side, 7 on the right-hand side, evenly spread out. Okay, should look, have I miscounted? That looks like 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Yeah, that's a bit of a mistake. So you should have 14 kind of evenly spread out on the other side, on each side of the cell membrane. Okay, so diffusion, passive transport has occurred. This is something we would do in a class, that I would get you all to gather on one side and then I'd ask you to diffuse. You know, feel free to do it with family members or you know, whatever, but it was just to do the same sort of thing. But you know, we would have set up the classroom and the tables in the middle as a cell membrane, but we can't do that. Now here is something you can do. Here's a nice wee experiment for you if you want to, something you can do at home. If you just get some Skittles and arrange them in that in a sort of circle like that, maybe on a plate, and just add a little bit of water, and what you'll begin to see is the diffusion of the colouring out of each of the Skittles, and you'll get a nice little sort of multicoloured plate in front of you. Okay, so you can try that if you want. So this is an experiment that we would have done in the class, and hopefully we can do it in the class at some point. So what we have is a model cell. So this here is around here. That would be the model of the cell membrane. And what you do is you put a mixture of starch and glucose inside. This is a bag. and after a period of time, you then test the water outside to determine whether or not the starch or glucose have been able to diffuse out of the cell. Now, what happens is starch is basically kind of glucose molecules joined together, and the starch is too big to get out of the cell membrane, so it will try to diffuse, but it can't get out, whereas the glucose is small enough to get through, so it will diffuse out. So what we would find with this is that you would, after a period of time, you would find starch inside the bag, glucose, but starch inside the bag, but not outside because it's too big to get out, whereas you would find glucose outside, it would diffuse out. Okay, press pause and have a go at this. So this is just a bit of a summary of what we were doing earlier on, see if you can remember. So press pause, and then when you're ready, press play and we'll go through the answers. Okay, the cell membrane consists of, and check your spelling on this one, number 1 is phospholipids, and they also contain proteins. It is selectively, 3 is permeable, meaning that only certain substances can pass through. And then diffusion is the movement of molecules down a concentration gradient from 5 higher to lower concentration. And just be careful with this one as well. You could think high to low, but it's not necessarily high to low. You know, it could be two very high concentrations. The importance is the comparison between them. So make sure you've got the R at the end, higher to lower concentration, until evenly spread out. And it is a type of number 7 passive transport that does not require energy. So this is the experiment we would have done in the class, so just to give you another little idea of it. So the starch molecule you can see is large, wouldn't be able, this is the model cell membrane, wouldn't be able to fit out, so it remains inside the bag, whereas the glucose is small enough to move out and diffuse outside of the tubing into the water, which is why you find glucose outside. So here it is. So this is what you would see after a period of time. Glucose gets out, but starch does not. Just an example here of some substances. So here's a cell membrane, the starch that we talked about, too big to get out, whereas a number of these substances can get in and out easily. Glucose, carbon dioxide, oxygen, and water. They can get through these channels or pores in the cell membrane. So let's just try and kind of move it on a level. We did it in the last lesson a few slides ago. You sort of did a kind of fill in the blanks task, and you put in one word answers and so on. So let's see if you can do this now from memory. So yeah, do it from memory, blank piece of paper, see if you can remember. So draw and label a cell membrane, explain what diffusion is, and then 3, explain why diffusion is important. Okay, so press pause, try all of them, and then come back. So let's have a look at each of these. So your cell membrane, as you've drawn it, doesn't need to be particularly detailed. The key part should be that you've got those kind of two layers of circles, and they're the phospholipids, and then you should have proteins, which are just kind of large blobs, which may or may not go all the way across the membrane. That's what you should have for number 1. Number 2, your definition of diffusion is when molecules move down a concentration gradient from higher to lower concentration until evenly spread out. You may have also mentioned that this is a type of passive transport. Then number 3, the reason why diffusion is important is in terms of biology and cells. It is because it allows important substances, for example oxygen, glucose, into cells, and waste products, for example carbon dioxide, to leave the cell. part of this topic is to look at a process called osmosis. So this is a type of diffusion, and hopefully from looking at that picture you can identify which type of, what specifically we're looking at here. So osmosis is when water molecules move from a higher concentration to a lower concentration through a selectively permeable membrane. So put simply, osmosis is the diffusion of water. So wee task here for you to do, so just follow the instructions, write down numbers 1 to 5, and what you're going to see is various pictures in which you've got a sort of membrane in the middle, you've got water on each side in different solutions and different concentrations, and what I want you to write down is whether you think the water is going to move left to right, right to left, or no movement. So number 1. So just to kind of give you an idea of the percentages, so the solution is 50% sucrose. That means it's half sucrose and half water. So hopefully you can work out which is going to be the more dilute solution, or which ones get more water, what way the water is going to move. So that's number 1. And you've got a higher water concentration on the right hand side. So imagine the right hand side, you could consider that to be 90% water if it's 10% sucrose. This side 50% sucrose, so 50% water I suppose. So you're going from right to left. Number 2. So you're going to have osmosis from left to right in this one, higher water concentration on the left hand side. 3. So this time we've got the kind of standard units for laboratory solutions, so molar. So we've got a higher concentration of sodium chloride on the left and on the right. Sodium chloride, remember, is just salt. So in this one, you're going to go from right to left. So the one on the right hand side, lower concentration of sodium chloride, so that means there must be more water, so it's more dilute, so it's going to move from right to left there. 4, 2 hydrochloric acid solutions. See what you think for that one. So this time the movement of water is going to be from right to left. Okay, so this is a lower concentration of hydrochloric acid, which means there's more water in it, so you're going to have water moving from right to left. And finally this one, so you've got pure water on one side, pure water on the other side, so you're going to have there's not much of a concentration, there's no concentration gradient there, so you're not really going to have very much movement of water at all. Okay. So related to this, I want to try a few little calculations. So if you've got a calculator, get it out, or get it open on your whatever digital device you're using. So hopefully you remember this from S3s or maths or whatever. Percentage change is equal to the difference in values divided by the original value times 100. So if you look at, here is an example of involving a jelly baby. So a jelly baby's mass changes from 6 grams to 6.3. So what is the percentage change? So if you look at in yellow, the equation we've got, the difference in values, well, it's moved from 6 to 6.3, so the difference is 0.3. The original was 6, that's what you started off with, so the sum you're going to do is 0.3 divided by 6 times 100. So the percentage change there is going to be 5%. Okay, so quite a common thing to do if you're measuring, you're looking at diffusion, you'll have water moving in and out of various things, and you can weigh them to work out whether or not diffusion has occurred. So next slide coming up, there's going to be a few wee calculations for you to do. If you can't remember that equation, you might want to just jot it down just now. So percentage change is equal to difference in values divided by original value times 100. So we're sticking with Jelly babies, 5 different jelly babies were weighed and then placed in different conditions. Calculate the percentage change in mass for each. So here you go, original mass and final mass for each of them, and I'd like you to give me answers for A, B, C, D, E. Okay, so press pause, and once you've done them, press play again, and I'll go through them with you. So the first one, it's going to be 5.6. So that's the difference. You've gone from 6 to 11.6. So 5.6 over the original, which was 6 times 100, which gives you a 93% increase. For the next one, you've got 5.8 to 5.2, so the change is 0.6. So it's 0.6 divided by 5.8 times 100, and that's a 10% decrease. For the next one, you've got an increase of 0.1. So it's 0.1 over the original 5.5 times 100, which gives you a 2% increase. For the next one, 6.2 down at 4.1 is a decrease of 1.9. So 1.9 over 6.2 times by 100 is a 34% decrease. And then the final one, you have got no change. Okay, so that's how you do percentage change. So this was a classroom experiment, a nice wee fun one, and again you can do this at home if you want. So basically all you do is you get Haribos. There are some types of Haribo which work better than others, and you just weigh them, stick them in water, and then you weigh them again afterwards. So if you feel like doing that at home, it's quite a good one to do. You know, if you've got a wee sister or brother, I think they'd quite enjoy this. So something you could try at home, weigh it before and after, and then you can work out the percentage change in mass. So what we're going to go into next is to explain what happens to animal and plant cells when placed in different solutions. This might be a point, if you've been sitting doing this really well now, you might want to have a break and press pause and come back and do this on another day. So we're just going to go into the last sort of section of this, okay? So we're going to look at particularly animal and plant cells. So again, this is an experiment you could do at home if you want, similar to the Haribo. I'll let you have a read of it, and you can. The directions are in the video, so you can go through it if you want. And basically, all you do is you get little slices of potato, and you weigh them, and then you put one of them in water, and then you put one in a sugar solution. So you could just, if you get sugar and just dissolve it in water, then you should be able to do this experiment at home. And again, it's just a case of measuring the change in mass over a period of time. Maybe you could leave it for, I think a few hours will be enough, to be honest. You could leave it overnight, and then you can do a percentage change. Here are the directions should you wish to have a go. So you would just change the sucrose solution, just put reasonable amount of sugar into some water and let it dissolve, and that would give you what you need for number 3. So this is the results of the experiment. So again, I'm hoping we can get in and we can actually we can do this. So if you've got the potato and it's placed in water, then you can see that over time it's going to increase. So it becomes bigger, heavier and firmer. So diffusion occurs, so diffusion of water into the potato, because there was a lower concentration inside the potato than outside. On the other one, you have got a very concentrated sugar solution. So that means actually in this instance, you've got more water inside the potato than outside it. So that means that water leaves the potato, it diffuses out, and you have the potato has shrunk. Okay, now what we've got here is an animation which shows what happens to plant cells when they're in water or when the water concentration outside is low. So at the moment you've got the cytoplasm, the concentration of water in here is the same as it is surrounding the cell. Now let's see what happens in various conditions. So if you lower the water concentration outside the cell, you can see what happens is water moves out and it begins to shrink in. So you can see the vacuole has become smaller, the cytoplasm is shrinking back from the cell membrane, and yes, water has left the cell by osmosis because the water surrounding the cell is less concentrated than the cytoplasm inside. And in these circumstances, we say that the cell has become plasmalized. Okay. If you go the other way, have a think about what might happen here. So you can see now cytoplasm is beginning to expand, vacuole is beginning to expand because water has entered the cell. So when water enters the cell and the vacuole expands and the cytoplasm expands, in these circumstances, we say that the cell has become turgid. Okay, cell has become turgid. And these two terms, turgid and plasmalized, we use them for plant cells, but not animal cells. Okay, so just a wee drawing of it here. So you've got equal water concentration, lower water concentration. So have a think, is that cell become plasmalized or turgid? That's a plasmalized cell. In the middle, normal cell, and on the right hand side that has become turgid. So if that was plant cells, let's have a look at animal cells. So this is an animal cell, this is a red blood cell, equal concentration of water inside and out at this point. So we go down to a lower concentration and you can see what happens is water has left the cell and it has shrunk. There's a word here, crenated, but we don't really talk about that in the course, so the cell has shrunk. If you increase the water concentration outside the cell, then more is going to diffuse in. What do you think is going to happen? Boom, it's burst. So for animal cells, they don't have that kind of protective cell wall. So what happens is if you just put them in water, they just become destroyed. They're burst. Okay? So difference there between plant and animal cells. So here we are, animal cells in water, left-hand side, you have got a shrunken cell, middle remains the same, and the right-hand side with a high water concentration, the cell becomes burst or destroyed. Okay, so that's osmosis in plant and animal cells. Just think about turgid plasmalized applies to plant cells, whereas animal cells in water will either shrink or burst. Now, diffusion we looked at as an example of passive transport, but we do have active transport. So you can probably work this out. If passive transport doesn't require energy, then active transport does. So if you look at the cell membrane here, what you've got here is more of the substance, in this case it's ions, outside the cell than inside. So what you would expect is naturally by passive transport these would move into the cell. However, if you have got energy, when membrane proteins use energy, they can move molecules or ions against the concentration gradient. So this can occur, the opposite of passive transport. So you can see despite you having to go against the concentration gradient here, if you've got energy, and this is ATP, which we'll talk about later in the course, then you can move a substance from a lower to a higher concentration. So sometimes you've got an example would be if you've got a higher water concentration outside the cell, you've got unicellular organisms which can use their vacuole to pump water out. Okay, but that requires energy, so active transport. Okay, wee activity here, so I'd like you to draw that, use plenty of space, and once you're ready, press play again. Okay, in the diagram, which is called a Venn diagram, what I'd like you to do is copy those into each section where you think it applies. Okay. Once you've done that, press play again and we'll see if you are right. So, active transport requires energy. It's from a lower to a higher concentration. It goes against a concentration gradient. And an example, K plus is potassium ions. An example is 0.1 millimolar potassium ions moving to an area with 0.2 millimolar potassium ions. For both, involves movement of a substance, occurs across a cell membrane. And then passive transport, the correct descriptions, does not require energy, is from a higher to a lower concentration, goes down a concentration gradient. An example is osmosis. Okay, so differences between active and passive transport. So we're just going to do a bit of a kind of a review of this whole topic. So have a go at this to begin with. So put down letters A to G, match them with the particular number, and yeah, press pause, come back, see if you've got it. So first one, A, diffusion, is number 2, movement of a substance from a higher to a lower concentration. B, osmosis, is 5, diffusion of water. C, active transport, is number 6, using energy to move a substance against a concentration gradient. D, passive transport, 4, diffusion is an example of this. E, plasmalized, is 3, when a plant cell shrinks. F, turgid, is when a plant cell becomes full of water. And G, phospholipid, the only one left, is 7, part of a cell membrane. Okay, next one I want you to try. So have a read through this, and there are 8 mistakes in here. So I want you to, I mean, if it was in the class, I'd be giving you a printout of this and you'd be circling it and so on. But yeah, you don't know. You might want to read through it and then just write down what the mistake is and replace the mistake with the correct word. Okay, so press pause and do that. There are 8 mistakes. So let's read through this. So cell membrane consists of phospholipids and carbohydrates. That should be proteins. And is selectively permeable, only allows certain substances through, e.g. oxygen. A concentration gradient is the difference in the concentration of a substance between two areas. Passive transport occurs down a concentration gradient from lower to higher. That's wrong, isn't it? So that should be from higher to lower, and does not require energy. Examples of passive transport are diffusion and osmosis. Next paragraph. Diffusion is the movement of molecules down a concentration gradient from a higher to a lower concentration until they are evenly spread out. This process allows important substances, e.g. carbon dioxide, into cells. Carbon dioxide you want out. So that's wrong, and we'll talk about what that might be in a minute. Carbon dioxide into cells and waste products, e.g. oxygen, to leave cells. No, that's not right either. So if you've highlighted them as incorrect, you could actually just have swapped them around. Another one you could have had going into cells would be glucose, or even water, I suppose. But yeah, you could just swap them. Osmosis is the movement of water molecules from a higher water concentration to a lower water concentration through a selectively permeable membrane. That is okay. Last paragraph. Animal cells burst when placed in water and shrink when placed in a concentrated salt or sugar solution. Plant cells become plasmalized, vacuole swells when placed in water, and turgid, vacuole shrinks when placed in a concentrated salt or sugar solution. They're round the wrong way, aren't they? So the first one, that should be turgid, plasmalized is wrong, and the second one, turgid, should be plasmalized. So how many we got? 1, 2, 3, 4, 5, 6, 7. So that means we've got one more in the last sentence. Active transport requires energy for membrane proteins to move molecules or ions down the concentration gradient. It's against or up the concentration gradient. I would say against the concentration gradient. For example, proteins which pump sodium out of cells. Okay, so that's a sort of summary that slide of what is required, the required knowledge for this subtopic. And finally, a wee bit of anagrams. So this is from this topic, so 1.2 transport across membranes, and also the cell structure topic. So remember the 4 different types of cell, fungi, bacteria, etc., and the different cell parts. So have a go at that, see if you can unscramble them, and then an appropriate sentence for each one. So press pause and do that. And I shall give you the answers. Number 1, chloroplast. 2, cellulose. 3 is plasmid. 4 is fungi. Sclamptoi, 5 is cytoplasm. Su-mis, osmosis is 6. Rit-dug, 7 is turgid. 8 is diffusion. 9 is phospholipid. 10 is vacuole. So if you haven't done so already, put down a sentence for each one. I'll give you examples of ones that would be reasonable. Number 1, the chloroplast is the site of photosynthesis in plants. 2, cellulose is a part of plant cell walls. Number 3, plasmids are where DNA are found in bacteria, or circular pieces of DNA in bacteria. Number 4, fungi is a type of cell with, you could go through the parts, you could do a drawing for that. You could say it's got a fungal cell is the same as like a plant cell but lacks chloroplast. 5, cytoplasm is where chemical reactions occur in a cell. Osmosis is the diffusion of water. Number 7, turgid is when plant cells become filled with water. They swell with water. 8, diffusion, hopefully this will be rolling off the tongue now. Diffusion is the movement of a substance from a higher concentration to a lower concentration down a concentration gradient. Number 9, phospholipids. Phospholipids make up a cell membrane along with proteins. And number 10, vacuole is a part of a cell which contains cell sap. You could say as well, when a plant cell becomes turgid, a vacuole swells. Right, that's it. That's National 5 1.2 transport across membranes finished.